job.
Thanks, Tim, for that nice introduction, and what a pleasure it is to be here. I've been here for a couple days now, along with all of you, for this great meeting, and it's really been wonderful to get to know so many of you, to know your names, to chat with you about your lives, your children, your relatives, your situations. And I'd really like to urge you to keep in touch. So you can find me on the web, you can find my email address, and please contact me if you have questions or you want to just chat a little bit by email. Please feel free. So I'm going to talk to you for about an hour about hereditary spastic paraplegia. And what I study is SPG4, but I think a lot of what I'll talk about is broadly applicable to various types of hereditary spastic paraplegia and maybe some of the other maladies that are suffered by people in the room. If you have any question along the way, just shout it out. Let's keep this informal. I don't have a whole lot of slides, so there's room in the talk for questions, and just shout out, raise your hand, and I'm happy to entertain questions intermittently in the talk. So just to start out, what is hereditary spastic paraplegia? Well, in the disease community, what I've learned is that diseases are usually defined by their symptoms. We cluster diseases by common symptoms, and then we kind of backtrack and look at the etiology, the mechanistic basis of the disease. So when clinicians label a malady as hereditary spastic paraplegia, these are the characteristics. It's really a characteristic gait defect, and I'm sure that in this short movie that the defect here is very familiar to almost everybody in the audience. This is really what it looks like when a person has hereditary spastic paraplegia. Of course, there's also plasticity, it's lower limb weakness. There are other symptoms. And usually we think about a disease as a simple case if these classic symptoms arise. But we call a disease a more complex case if there are other symptoms. So in different patients with hereditary spastic paraplegia, there may be other symptoms. There may be some upper limb problems. There may be some cognitive difficulties. So it really all depends on the individual person level. But here's how hereditary spastic paraplegia is generally defined. Now, in most cases, it's adult onset. It's a rare disease. When I say most cases, we're not talking about a whole lot of people. But what I've come to learn really in recent years, and talking with patients in particular, is that it's really not adult onset in a significant number of cases. I've really had the opportunity to work recently with some parents of very small children who have hereditary spastic paraplegia, and also I've chatted with some parents here in the room whose children are maybe 10 or 13 or 16 who are showing some debilitating symptoms, or at least early onset symptoms. So it's usually adult onset. I think usually the age where symptoms become apparent, maybe mid-40s, but there are a lot of people who have symptoms in their 20s or even earlier. And there is an enormous amount of variability among patients, even within the same family. People who have the same mutation to the same gene in the same family. Some individuals in the family may be in their 60s before they really detect any sort of symptoms, whereas other people in that same family might be 4 years old, 4, 6, 8, 20 years old, any number of ages. And this is really part of the mystery. Why is there such a big disparity, such a big range of age of onset? A little bit of a part of it is that some people may have symptoms, but they don't want to admit it to themselves or their doctors that they're suffering some sort of patients. Usually the spouse is aware, though, that some symptoms are early on. That's part of it, but other parts of it we don't completely understand. It may be that some individuals are just better able to compensate for the presence of mutant proteins in their system. But that's really one of the questions that we'd like to understand better. Anatomically, what is hereditary spastic paraplegia? Well, there is something in your spinal cord called the corticospinal tract. And these nerves originate from your brain. So in your brain, there's something called the motor cortex. And each of these nerves have what's called a cell body. That's where the DNA is, the nucleus, the DNA. That's located in your brain, in the motor cortex. And then those corticospinal motor neurons extend their axons, their nerves, also called axons, collectively down through your spinal cord to form what's called the corticospinal tracts. It's not the only tract in the spinal cord, but it's one of the tracts in the spinal cord. And this is really important for the motor capabilities of the limbs. It's believed that in hereditary spastic paraplegia, what happens is there's something called a dying back degeneration, where these nerves detach from their synapses, their connections, and then they're going to slowly pull back from the synapses. And once they pull back, they don't have their connections anymore, and therefore they won't work. So this is what's believed to happen. And along the way, there are some pathological swellings of the axons that will also indicate that something's wrong. So corticospinal degeneration is really the principal anatomic basis of hereditary spastic paraplegia. But as I mentioned, some cases are what we call complex. And there are other tracts in the spinal cord, other parts of the brain, that may suffer from some degeneration. So principally, it's corticospinal tracts, but let's not get too preoccupied with corticospinal tracts because it could be other things as well. The other thing I want to point out that came out in the discussion yesterday was that we really have very little human tissue to look at what's happening with the patients. So I think Dr. Fink said yesterday that we have about 12 corpses that the anatomy has been examined in patients with hereditary spastic paraplegia. So everything you see on the slide and everything that I've said is really knowledge gained from a very limited number of corpses, people who passed away whose anatomy has been examined. So there's really a lot to learn, but this is essentially what we know for now. Hereditary spastic paraplegia, as I said, is a cluster of different people who have similar symptoms, the gait defects, the spasticity. What we've learned in recent years is that there are several genes whose mutations give rise to what we call hereditary spastic paraplegia. Now, is it all the same disease, or are these all different diseases that manifest similarly? And this is something, again, we don't quite understand. Now, when we talk about the genes whose mutations give rise to this disease, they're quite different from one another. They're quite different from one another, which might suggest that these are really different diseases. But on the other hand, maybe not. I mean, maybe what's wrong with these genes, the mutant proteins that they produce, maybe they're tapping into common pathways, maybe they're tapping into the unique vulnerability of the corticospinal tracts. So it's possible it's all the same disease, but it's also possible these are actually very different diseases that look similar when we look at their symptoms. When I first started studying hereditary spastic paraplegia around 15 years ago, I think there were 12 genes that had been identified in patients. Now, if I'm not mistaken, I think there's 92. So it's a really growing number of genes whose mutations give rise to these symptoms. Interestingly, I think I'm correct when I say that still the majority of patients who are diagnosed with HSP don't have a mutation in any of these 92. We're still looking. We're still looking. There are still patients who fully manifest as having HSP, but we don't know which gene is mutated yet. Of the cases that are diagnosed, I think about 40% have mutations in a gene called Spast. Spast used to be called SPG4, and this type of hereditary spastic paraplegia is called SPG4 HSP, or sometimes just type 4. And that's the type that I study. It's the most common. I think of the people in the room who know what gene is mutated, it's more likely SPG4 than any other gene. What's interesting, though, is it's not just one or two different mutations in Spast that give rise to the disease. It's really over 200 different mutations that give rise to this disease. And these mutations can arise in all different parts of the gene. There's not one special part of the gene where the mutations are particularly pathogenic. It can be in all different parts of the gene. And this is really interesting because I think what it's saying is that this particular gene is highly vulnerable to give rise to pathology. And I think that's really interesting in telling us something. Now, I want to talk just a little bit about what I mean by mutation, because I know a lot of you, this is your very first meeting, and maybe some of these terms are a little fuzzy. So inside all of your cells, there are chromosomes. And humans have 46 chromosomes. So we have 23 pairs, one from your mother and one from your father. Now, you all know about the XY. Men are XY and females are XX. All of the other chromosomes are identical, though. You have one from your mother and one from your father, and they're called your autosomal chromosomes. You have the sex chromosomes and then the autosomal ones. And the Spast gene is an autosomal gene. So it's not present on XY, it's an autosomal gene. And the inheritance is autosomal dominant. So I think most of you have heard about recessive and dominant. If it were recessive, if the mutation were recessive, that would mean that you'd have to have the same mutation from your mother and your father, otherwise you're okay. But if it's dominant, it means all you need is the mutation from either parent, and then you've got the disease. That's what we mean by dominant. What we mean by mutation is the DNA sequence of that gene has changed. Somehow it's changed. This actually happens all the time, that mutations happen, you have the DNA code, and maybe one of the aspects of the code just switches. Sometimes it's actually just randomly switches. And this happens all the time. And almost always, it's harmless. Almost always, it's harmless. But every once in a while, one of these little switches, these little mutations that happen, it's not harmless. It causes disease. So when we talk about pathogenic mutations, that's what we're talking about. So this somewhere, if you've got hereditary spastic paraplegia in your family, at some point along the way, a grandparent, great-grandparent, great-great-great-grandparent, someone had a spontaneous mutation in the Spast gene, and that caused hereditary spastic paraplegia, and that was passed on through the generations. There are still people today who can have a spontaneous mutation. So there may well be some of you in the room who you yourself or maybe your child has hereditary spastic paraplegia, but nobody in the family had it before. Well, how can this be? Well, that's what happens. The spontaneous mutations happen all the time. And one of the things I found quite interesting is that usually when it's spontaneous, it seems to hit these children very young. So of all of the children, the parents that I've interacted with, it seems like it's always a spontaneous mutation. It seems like the parents don't have it, or it's not in the family. And what exactly that's telling us, I'm not quite so sure, but one possibility is it's telling us that if it's hereditary in your families, that means that your genetic background is accustomed to having it and kind of maybe adjusted to having it so that you're okay to have your own children and pass it on. But these small children who acquire what's called a de novo or spontaneous mutation, their genetic background's not used to it, and it's going to hit them very, very hard. And it's the same mutation that adults have. It's not a different mutation. So that's one of the mysteries that we're working on. Another thing is the confusion with other diseases. If you can't read this writing, it's too small, don't worry about it, I'll talk you through. The other is confusion with other diseases. So I had a phone call a while back from a mother from Michigan, and her son is 16 years old, and he'd always been diagnosed as having cerebral palsy. And then it was discovered, no, he has a mutation in the Spastic gene. And this was a case where it was a complex case where he also has some cognitive difficulties, and it was always viewed as cerebral palsy. So when we think about hereditary spastic paraplegia as a rare disease, and I think Dr. Fink said maybe about 25,000 Americans have it, it could be a lot more. It could be a lot more because there's a whole lot of misdiagnosed cases. Now let's talk about Spast. This is the Spast gene. And what's really interesting about the Spast gene is it has what we call two start codons. And all that means is that the gene can start coding for the protein at one place or a different place. And so it'll create a long protein or a little shorter protein. Now, keep in mind, that's what genes do. Every gene encodes for a protein. That's what genes do. There's always a protein, and proteins have different functions. What Spastin protein is an enzyme. And enzymes hydrolyze ATP, they use energy, and they get some kind of work done for the cell. So the longer protein is called, we call it M1 spastin. And the somewhat shorter protein we call M87 spastin. M87 spastin is quite widespread. It's in every cell of your body, it's quite widespread, and it's abundant. M1 spastin is somewhat more low quantity. It's harder to detect, there's not as much of it. And what I'd like you to think about is that these two proteins are quite different. And I think Dr. Blackstone likes to describe it as two different proteins. So M87 protein is a microtubule severing protein, and M1 is a protein that regulates endoplasmic reticulum and membrane structures. So we'll talk a little bit more about that in a moment. But the big issue here is that one of these is in high abundance, the other is in low abundance, and they really do very different things. Now, taking a look at what they do, here's a neuron, and I think Honda showed some nice pictures of neurons earlier, and you can see there's that cell body that I talked about. It's got the nucleus, that's what the DNA is. And if you can see, I know the image is very small on the screen, but if you can see that elongated extension here, this is the axon. That's what's going to get very, very long and extend into the spinal cord. inside that axon, you have these structures called microtubules. And what microtubules are like the 2x4s in a building. They're the architectural elements. They also serve another function, and that's as a railway. So most things are manufactured here in the cell body, then they need to move down the axon over these very long distances, and sometimes back the other way to the cell body. And how that happens is to move along these microtubules. So think of them in two ways. One as the architectural elements that allow the neuron to take on this very interesting shape. And the other thing microtubules do is to serve as railways. What you see here, these are called molecular motor proteins, and they're what move along the microtubule to convey different sorts of things. What's depicted here is called the endoplasmic reticulum, and that's a network of membrane, of internal membrane. So M87 has its big role in cutting microtubules. That's what you see here, severing the microtubule. Now, before you panic, that's not a bad thing. Severing microtubules is actually a physiological, important, normal thing that happens. Not too much, because you don't want to break your microtubules to nothing, but breaking microtubules into shorter pieces gives them greater mobility and greater ability to move around and underlie the plasticity of the nervous system, which is really important, even in the adult, that your nervous system is plastic. The endoplasmic reticulum, as Dr. Blackstone has pointed out, is really important for M1. M1 inserts into the ER, and if Dr. Blackstone specifically gets rid of M1, he sees some major changes in endoplasmic reticulum. So it's important for shaping the endoplasmic reticulum, and also some other membranous structures called endosomes. Now, this is what these proteins normally do. And what I want you to do now is think a little bit about loss of function and gain of function, because this is really what disease is all about. Sometimes, if a protein is mutated and it doesn't work as it normally should, that's why you have a disease. Insufficient microtubule severing or insufficient regulation of endoplasmic reticulum. That could be why you have a disease, because that protein normally has work to do, but you've lost that ability to do that work. Now, remember, you have two genes. One's still okay, so the other is not. So you have maybe around half as much. So sometimes that's called haploinsufficiency. Half is not enough. That's one way that you can have a disease. The other way is called gain of function. Now, gain of function sounds like a good thing, but it's not. Gain of function, maybe I should call it gain of toxicity, because what that means is that mutant protein is still there, it's not doing what it should do, but it's causing some sort of trouble. And it's usually because a protein misfolds. So that protein changes its shape, misfolds. Imagine you should fold your towel one way, but you folded it backwards or something. And what this usually does is it exposes domains on that protein that should be hidden. So maybe you have a domain that's important for activating something or other in the cell in certain discrete circumstances, but now it does it all the time, and it's too much. So that's what we mean by gain of function. And it turns out in neurodegenerative disorders, gain-of-function toxicity is actually more common. It's usually a more common mechanism. There are some diseases that really are caused by haploinsufficiency, just not enough of that function. But other diseases, say for example like ALS, it's really what causes it is the mutant protein misfolds, and now it's really doing bad things. And if I could just illustrate this, here's loss of function, is like the kid didn't show up for class. He's simply not there. That's not good. But gain of function, oh, he's there all right, but now he's causing a lot of trouble. And that's the gain of function. And it gets worse. And there's one bad kid can cause a lot of bad kids. And so that's kind of a way you can think about loss of function and gain of function. Now, M87 is detectable everywhere. And I usually show some what are called immunoblots or Western blots showing this. But I know you're not scientists, so take my word for it. M87, everywhere we look, there it is. M1 is present in such low levels that we really don't see it anywhere. It's there, but we don't see it because it's present in such low levels, except for the one place we see it is in the corticospinal tracts. So that within itself gave us a bit of a clue that maybe it's M1 that's the troublemaker. M87, well, if it doesn't work right, that's probably not good. But M1 may be really the troublemaker because it's present at higher levels in the corticospinal tract. So what kinds of difficulties might spastin mutants be causing? So anything I just talked about, microtubule severing is not going to be quite as active as it should be. The ER might not be quite as normal as it should be. But what about gain of function? Now, with gain of function, it could really be almost anything. When a protein is misfolded and domains are revealed that should be hidden, a lot of times what those domains will do is they will hyperactivate a kinase or some other sort of enzyme. Now what a kinase is a protein that puts a phosphate onto other proteins. And that turns out to be really, really important. Say, for example, with these motor proteins. You start phosphorylating those motor proteins more than they should be, and they won't work right. And they won't move along the microtubule properly. They won't convey their different cargoes properly. There are different sorts of things that these gain-of-function properties can do that you would never predict, And that, I think, really becomes at the heart of hereditary spastic paraplegia to attempt to understand what it is these mutant proteins are doing. Now, maybe the mutant proteins in the other forms of HSP are doing similar things in terms of gain of function, even though they have their normal protein functions are very different. That's one possibility. So over the years, I've used different experimental models with different collaborators. So a lot of what I've done is in the culture dish, meaning that I culture different kinds of neurons and investigate how they grow in the culture dish. And again, Honda talked a bit about this earlier. But other sorts of systems that I've used with collaborators, one is squid. Now, you may think, squid? Why would we be using squid? It turns out that squid have something called a giant axon. It's a huge axon that evolutionarily came about by the fusing of multiple axons. So you can squeeze out that contents of that nerve like toothpaste, and then you can really put on all kinds of different proteins and inhibitors and all that and study it. It's a beautiful, beautiful system. We've also looked at Drosophila. Drosophila is the common fruit fly. And you can very easily manipulate the genes of fruit flies. So whereas with a higher vertebrate animal, it would take like $50,000 to create every new kind of mouse. With flies, you can generate as many flies as you want quickly, and the generations are very short. So it's a very good experimental system. What we found with all of these different systems, when we took mutant M87 and mutant M1 separately, and we introduced them into all of these systems, every time the mutant M87 doesn't do anything bad. The system tolerates it just fine. Every time, in side-by-side comparisons, the mutant M1 creates bad problems. If you express the mutant M1 in the fly, for example, the fly can't fly anymore. You put the fly on its back, it can't turn itself around. The fly can't climb properly. That's with mutant M1. You put in mutant M87, the fly is pretty normal. With a squid, you can just insert the protein. You can simply put in mutant M87, and you can see that everything that moves along the microtubules is fine. You put in mutant M1, and now what moves along the microtubules doesn't move anymore. So it seems that it's the M1 protein that's really prone to toxicity, and it doesn't really matter what the mutation is. Whatever mutation it is seems to cause this kind of misfolding. And if you think about evolutionarily, maybe this makes sense. Why do you have two start codons? One start codon is very good. That's the one with M87. The one with M1 is kind of not as good of a start codon, an imperfect one, and that's why you have so very little M1. But evolutionarily, we had to hold on to M1 because it's important in very small amounts for the ER. But that M1 protein is like dynamite. Anything that goes wrong with that molecule is going to open the door for this toxicity. Now, what we really need is a higher vertebrate model that we can study gait defects, and we can test therapies. So it was really important that I move out of these different invertebrate models and move into a model that really has a cortical spinal tract. So we moved into genetic mice. This is the most common system that people use for looking at vertebrate diseases. Interestingly, in the HSP field 15 years ago, when I first got involved, it was the common view, almost the universal view, that the disease was entirely caused by haploinsufficiency. So when people studied this disease, they used what are called knockout mice, where they simply removed one or both of the Spastin genes. And then they said, well, this is my model for hereditary spastic paraplegia. The problem with that is that those mice have no gait deficiency. So I think all along that was telling the community something that everybody needed to appreciate, and that's that haploinsufficiency is probably a component of the disease, but it's really not the principal component. The principal component is this gain of function toxicity that I mentioned. So what we did is we took this mouse, here's the wild type, and you can see that mouse moving on this rotorod. If I took the knockout mouse that has no Spastin genes, if I took the knockout mouse and I put it on that rotorod, it would look pretty much the same. It would look the same. So why were people using the knockout mouse as a model for HSP? So what I did is I took a mouse and I introduced mutant human spastin. I left the mouse genes alone. They're still there. So this animal that I'm going to show you in a moment is not haploinsufficient. It has all its normal mouse genes. The only other thing it has is human mutant spastin. Now here's this mouse. It has a lot of trouble walking. It's kind of scrunched up. And it clearly has the symptoms of hereditary spastic paraplegia. So it's still a little controversial. There are still people in the field who like the idea of haploinsufficiency for whatever reason. But I think that this mouse is the best evidence of all, that you don't get the symptoms of HSP with haploinsufficiency, but you do get the symptoms of HSP with gain-of-function toxicity. Now, when I take the knockout mouse and this knock-in mouse, and I cross them, so now I've got a line of mice that are both. The symptoms are worse, earlier and worse. So I think what that's telling us is that haploinsufficiency is not the primary cause of HSP, but it's an exacerbating factor. It's an exacerbating factor. It's making the corticospinal tracts more vulnerable to the mutant protein. And that's actually a big deal, because it may be that this helps explain why it is in the same family some individuals may be okay for many decades, whereas other individuals in the same family are not. Because really the haploinsufficiency component is involved in setting that clock. Now, what I'm showing you here is we've taken this mouse and we've crossed it with a fluorescent mouse, similar to what Honda was telling you about. And this is the brain of the normal mouse. And these are corticospinal motor neurons, just like Honda was telling you about. And this is a higher mag view of them. And they're fluorescent. So what's nice about this is that all we have to do is take this mouse and section the brain, and we can see the corticospinal motor neurons. This is the mutant mouse, and this image is from a symptomatic mouse. So this is about 3 months old, and it's showing the symptoms. And what I want you to appreciate here is you just see fewer neurons. You just see fewer neurons, maybe less than half. So this is really quite a nice illustration of what's going wrong. And I think also what this mouse is going to enable us to do is to really test therapies much more rapidly than if we had to take every potential therapy and do a lot of tedious behavioral work. We can rush the work much quicker by simply looking and seeing if we've rescued the number of neurons that are present in these symptomatic mice. A recent finding that we've just begun to see, so it's very, very early, but we have looked pre-symptomatically at a mouse that's about 1 month old, and now that mouse does not yet show any gait deficiencies, and yet you can already see the loss of these corticospinal motor neurons. And I think that, again, that's very, very important, because before we saw that result, I would have thought that until you start showing symptoms, your corticospinal tracts are probably fine until you start showing symptoms. But no, what these results suggest to me, and again, they're very, very early, is that people who are perhaps even decades before they show symptoms, that their corticospinal tracts are already showing this level of degeneration. How they're somehow able to be okay in the face of having so many fewer corticospinal motor neurons that are healthy, I don't know. But that's a good question, and maybe one avenue toward helping other people get by effectively with fewer neurons that are normally wired. So I know you can't see this slide, it's too small, but what I'm hoping to illustrate here is that there are really two components of the disease, SPG4 HSP. There's the haploinsufficiency component, which again I think is an exacerbating factor, and then there's the gain-of-function toxicity of the mutant protein, which I think is the primary cause. But either one of these, if we can find ways of treating them, of making it better, either one of these can help patients live normal lives, theoretically, for decades. And maybe that's all you need, is a few decades more. So we're really interested in understanding the underlying mechanisms. With loss of function, I think it's a little clearer. Not enough microtubule severing, not enough membrane regulation. So how can we compensate for that? Well, I've studied microtubules for 30 years, and Dr. Blackstone has studied endoplasmic reticulum for probably equally as long. So we have a lot of knowledge about these that we can utilize to help repair the microtubule system and the ER system in these cells. That said, though, the gain-of-function toxicity that's really the more important cause of the disease, that again gets really, really tricky. We've done some work where we think that part of the toxicity is a hyperactivation of a kinase called casein kinase 2, CK2. And this is what we think is causing those motor proteins I mentioned to become inactive, is that this kinase is too active, it's causing a lot of phosphorylation where phosphorylation should not happen, and that's a problem. Well, so maybe we can think about casein kinase 2 inhibitors as a potential therapy. One of the targets of CK2 is another enzyme called HDAC6, and that's really important for adding what's called an acetyl group to certain proteins to help regulate them. So maybe inhibiting CK2, that's too much. If you inhibit CK2, maybe that's going to cause too many problems. So let's go down a level and let's inhibit HDAC6 and see if that's better. So we have different levels that we can go, and these are just two of the things. There may be a lot more different targets of its toxicity that are even more important. But you can see that these are two avenues by which we can start thinking about pharmaceuticals. Gene therapy, I think, is probably one of our best bets. And I know you've heard about gene therapy already, and it's quite exciting. And I'm working with a gene therapist named Miguel Estevez. And the whole idea of gene therapies, I think most of you probably know, is what we want to do is we want to turn off the mutant gene and replace it with a normal gene. So this is a matter of coming up with a certain kind of viral vector. It's driven by a virus and it gets into your system, and we put into there the DNA for a good spastin gene, and then we use something called microRNA that knocks down the mutant gene. That's the whole idea. And we get it into your nervous system, and slowly you're going to stop making the mutant protein, and you're going to start making more of the normal protein. Now, there are complications to this, one of which is we want to be really, really careful not to make too much normal protein, because you make too much spastin, you'll chop up your microtubules to nothing, then you'll really be in trouble. So we had to be careful. There are other sorts of things we need to be careful about as well, or at least aware of. Now, let's say for many years, your spinal cord has been accumulating misfolded M1. And we come along with gene therapy and we turn off the expression of mutant M1. Well, you've still got a spinal cord that's full of the mutant protein. It's still there. Now, usually misfolded proteins are degraded by what's called the autophagy system. But the autophagy system is also going to be depressed because it relies on the microtubule system. So it's really a tough situation. We may have to combine therapies where when we treat a patient with gene therapy, we also have to treat with one of these drugs. Or, in addition to what I talked about before, an autophagy enhancer to make the autophagy better. So it may really be a combinatorial therapy at the end of the day, what's really going to work. I want to mention real quick that the way that I met Dr. Estevez was that there are a group of parents who have small children with HSP. They discovered Dr. Estevez, they discovered me, they put us together, and they are not scientists, but they did all of this, and they really are to be applauded. They have their own foundation called the SPG4 Foundation, and these parents are absolutely wonderful in their determination to make things happen. So that's really, really noteworthy. There's some other things that we can do. CRISPR-Cas is a very new method for gene therapy where you're actually going into the genome and you're not depleting and replacing, but you're actually trying to fix the mutation. That's kind of the newest thing. And then the other way, and I think someone mentioned it earlier today, is antisense oligonucleotides. And what antisense can do, we're working with a pharmaceutical company called Ionis, and they're the best. They're incredibly good at antisense. And what you can do with the antisense is you can actually target the mutant Spastin gene, and you can leave the wild type alone. You actually make it to target the mutation. So what this therapy is going to do is going to get rid of the mutant protein, or stop its expression, but it's not going to replace it. It's not going to replace it. So it's not going to solve the haploinsufficiency part of it. But my guess is that's probably okay. It's probably okay. You can probably get by with one Spastin gene. But what's really nice about this antisense approach is that there's no virus involved in order to get it to work. It's just simply injected, and it lasts for a few months, so you'll have to go in and have multiple injections. But the good news is if you have a bad reaction to it, it'll be gone before you know it. And I think that's a big asset. So these are the sorts of things that are going on that I think are exciting. And again, with the drug therapy, CK2 inhibitors, HDAC6 inhibitors, and autophagy enhancers that I mentioned before, and different loss of function avenues we can take exploiting our knowledge of that. One of the other things I think that's a really good idea that I like is instead of going in with CK2 inhibitors or HDAC6 inhibitors, because these are enzymes you need in your body, you don't want to inhibit them too much, is can we find inhibitors that will actually interact with that domain, that M1 specific domain, and block it? So even if your spinal cord is full of this stuff, we put something in that blocks it, that blocks its toxicity. That should work pretty fast if we can find something. And I'm pretty excited about that possibility. I want to just acknowledge people who have worked on this. So Gerardo Morfini is a partner of mine. He's at University of Illinois at Chicago. He's done the squid work, and he's also my collaborator on the fluorescent mouse work, and so he's an incredible guy. And then Liang Qiang was my former graduate student, and he worked on Spastin in my lab when he was a boy, and he's returned to Drexel University. He has his own lab, and he's an incredible guy with his own interests in HSP, and I hope he can attend a future meeting because you'll really, really like him. Emanuela is our postdoc, and she's from Italy, and she's been working on the mouse from the beginning, and she's also incredible. So I also want to acknowledge the Cure SPG4 Foundation, these wonderful parents that I mentioned earlier, and their efforts. Of course, the SPF Foundation that are doing such incredible work here, and I want to thank you for your very kind attention.
Dr. Baas, would you like to take a question or two?
I would love to take
You would love to.
questions.
Well, since I'm up here, this is where we're going to start.
So, like with gene editing, like with CRISPR, say, like I understand how it would work in an embryo with only one or a few cells, but how would it work in an adult with trillions of cells?
Yeah, so with the gene therapy, you would introduce it either into the motor cortex or the spinal cord, and so it can be transported up to your brain. But you would not introduce it into every single cell of your body, but you'd target it for the motor cortex. I think one of the things that's possible is to actually put the vector on a promoter that's specific to corticospinal motor neurons, and I know that Honda's doing a little bit of work with that, and that might target it more specifically, the neurons in question. But once it gets into the adult, presumably what'll happen is you will stop the progression of the disease. But then the next question I'm sure that you're all very invested in is, how do I get better? How do I get functional recovery? How can I walk again? And these are very, very ambitious goals. And they are certainly, though, on the radar, that simply stopping the progression isn't good enough. We have to also think in terms of how we can promote regeneration of a corticospinal tract that has already regressed, or how can we promote sprouting of neighboring axons that can take their place. Fortunately, at Drexel University, I'm part of the Spinal Cord Injury Center. We have a Center for Spinal Cord Injury, which deals with people who have damage, injury. They jump in a swimming pool and they hurt their spinal cord. And that's what they do, is regeneration and sprouting. That's what they do. So I'm hoping to tap into their knowledge that once we stop the progression of your disease with the gene therapy, of how we can get functional recovery through regeneration.
Okay, another question. Oh, it's over there. Okay.
Very interesting, thank you. So for CK2, if you have SPG4 and you have a lot of it, and the hex, whatever it is there anything that people may be taking now, either for other medical issues, that could promote CK2, which you probably don't want based on what you say there? And if so, is anybody looking into things that you probably shouldn't be taking as medicines, or probably you shouldn't be doing because it might promote CK2, which is not good?
Yeah, I would not overreact to that too much. I mean, remember that there's not very much M1 in any other cell of your body. It's really the corticospinal tracts that have more M1, and they're more prone to this toxicity. So in the corticospinal tracts, there's going to be more of this hyperactivation of CK2. And these days, the inhibitors of CK2 or any kinase are very dirty drugs, and they inhibit a lot of different kinases. They're not very specific. And so, and also CK2 has a lot of important functions. So it's not immediately on the radar to use CK2 inhibitors to treat the disease. Your point, which I think is a very interesting one, is do you want to be a little careful not to make that worse by other sorts of drugs that you might take? And I'll investigate it for you. I'll poke around a little bit and see what I can find out. But at present, I wouldn't worry about it too much.
Let's do one more. One more. Wow, far out, no hands. This is great. Okay, perfect. Great job, Dr. Baas. Fantastic.